Hello dear students, welcome back again to Chinmoy's Biology channel. So today we are here with a brand new episode of the fundamental unit of life. So this is a very important topic in biology and hope you will be enjoying this lesson with me. So let's get started without further delay. So today we are starting with the chapter 5, that is a very important lesson. This is fundamental unit of life. So we all know, we have already studied about cells. So let's see what we are going to study today. So while examining a thin slice of cork, what did Robert Hooke notice? He noticed that the cork resembled the structure of honeycomb because it is having many little compartments in it. So cork is a substance. What is cork? It is a substance which is obtained from bark of a tree. So this happened in the year 1665 when Hooke he made an observation through a self-designed microscope, right? Robert Hooke called these boxes as cells. So cell is a Latin word for a little room. So what does this cell mean? This word cell means a little room. Is it clear? So Robert Hooke saw that in this cork there are honeycomb-like structure inside and they are having small compartments. So these compartments were told by him that these are cells. Now this may seem to be very small and insignificant incident, but it is very important in the history of science. So this was the very first time that someone had observed that living things appear to consist of separate units, right? So the use of the word cell to describe these units is being used till today also in biology. So let us find what are cells, right? So what we are going to study today, we are going to study today about what are the living organisms made up of, what is a cell made up of, and what is the structural organization of a cell. So bit details introduction we are going to learn today, and in the next day we will go to the full details of this lesson, that is the fundamental unit of life. So you need to subscribe to my channel, stay tuned, because when I update the next part of this lesson, you need to get the updates and you need to complete the lesson in that way. Right, so stay tuned, stay updated. So let's start with what are living organisms made up of. So first we are going to start with this activity. So this activity generally you are doing in the class or in the lab. So let us take a piece of onion bulb, and with the help of a pair of forceps we can peel off the skin. So the skin of the onion cell, this is called the onion bulb, is called the epidermis. Okay, and from the concave side, the inner layer of the onion, so you are peeling the skin from the concave side, that is from the inner layer of the onion, right? So this layer, it can be put immediately in a watch glass containing water. Right, so this will prevent the peel from getting folded or getting dry. So you are peeling the epidermis region of onion and you are putting it on a glass slide, and you are putting it on the glass slide which contains a little bit of water to prevent this peel getting folded or getting dry. So what do we see in this peel? So let us take a glass slide, put a drop of water on it and transfer the small piece of peel on it from the watch glass to the slide. So first we are taking it in a small watch glass and then we are transferring this on the glass slide. So make sure that the peel is perfectly flat on the slide, and the thin camel with a they are telling with a thin camel hair paintbrush or might be something necessary to help to transfer this peel from the watch glass to the slide. So what else you can do? You can just take delicate forceps. With that you can just take the part of the peel and spread it evenly on the piece of slide. Now you put a drop of saffron in solution on the piece of that and you cover it with a cover slip, and the cover slip should be placed so minutely that no air bubble should be there while putting the cover slip. And this mounting should be done with the help of a needle. So if you are not holding a needle at one end of the cover slip, then it's you will end up in putting some air bubbles inside that cell, okay, the epidermis or the onion peel. So now ask your teacher to help you. We have prepared a temporary mount of an onion peel, and we can observe this slide under the low power microscope and high power of a compound microscope. So what do we observe? Now you know that this is a compound microscope and these are the parts of a microscope. This is very important for you, and just go through this diagram with the label parts, so that if in your exam you get this diagram with the label parts, you can write it. So what is this? This is the body tube. This one is the eyepiece, that is through this part only you see through it. This is the coarse adjustment screw, and this is the fine adjustment screw. And once you go in your lab, you can just see a compound microscope and match the parts with that. So this is the arm. This arm only you are holding to take the compound microscope from one place to another. So this is the objective lens. So first you are seeing in the low power, then you are observing in high power. So this lens only you are observing. Now this is the stage on which you are putting the glass slide. You are mounting the things which are to be observed are put on this stage. These are two clips on the two sides. So these clips allow you to hold the slide in its position on this stage. Now this is the swivel, okay, and this is the mirror where the light falls and through which you see this you see this objects in a magnified manner. And this is the base of the microscope, and this is the condenser. So this is full compound microscope. So we have prepared onion peel now. So we are observing that under the microscope low power first, and then we are setting the power to high power. So what do we observe? Let us see here. Now this is what we are observing. We are observing this under the microscope. We can see this like this cells. We can see one nucleus, and we are seeing this cells. So what does it look like? You can just take a sheet, white sheet, and you can just draw this onion peel. So we can try preparing temporary mounts of peels of onion of different sizes. And what do we observe? So do we see similar structures or different structures? So we just need to observe a few peels, and we just need to observe them under the microscope with just one drop of saffron in solution. So then we will see the nucleus, the cells, and different shape and size of the cells. So what are these? We need to draw, and we need to write the different structures, right? So what are these structures? What are these structures? These structures look similar to each other. So they form a big structure like an onion bulb. So we find from this activity that onion bulbs of different sizes have similar smaller structures visible under the microscope. So these similar smaller structures are called cells, regardless of their size of the onion they came from. So whatever onion you are taking, whatever onion you are peeling the epidermis part and putting it under the microscope, you are seeing that there are small boxes of things present in the onion peel, and these are called cells. That is, they are like small boxes or small rooms. So these same structures that we see, okay, so they are the basic building units of the we can say that these are the basic units of the onion bulb, and these structures are called the cells. Not only the onions but all living organisms we observe are made up of cells. However, there are also single cells that live on their own. So we are all multicellular organisms. That is, our body is made up of many cells, and there are also organisms which are made up of only single cell. So cell, we have discussed that it was first discovered by Robert Hooke in 1665. He observed that the cells in a cork size slice, and with the help of a primitive microscope only, he told that cells are present. Now in 1674, Leeuwenhoek he came, and with the improved microscope he discovered that free living cells in pond of water for the first time. So it was Robert Brown in 1831 who discovered the nucleus in a cell. So first it came to us in 1665. Then Leeuwenhoek in 1674 improved the microscope, discovered free living cell in pond. Then in 1831 nucleus was discovered. Now 1839 protoplasm it was discovered as a fluid substance present in the cell. So the cell theory that all the plants and animals are composed of cells and cell is the basic unit of life was presented by two biologists, that is Leiden and Schwann. So Leiden and Schwann in 1838 and 39, they proposed the cell theory. So cell theory was very important, and it only proposed that cells are the basic unit of life, and this cell theory was further expanded by Virchow in 1855, and by suggesting that all cells arise from the pre-existing cells, right? Now, with the discovery of electron microscope in 1940, it was possible to observe and understand certain complex structures of the cell and its various organelles. So it came from 1665 till 1940. The process was a continuous process that one after the other got invented. So this is what we should know, that how this cell theory came into being and how it was gradually, after several experiments in 1940, it was possible to observe the basic complex structure of cell and its organelles, right? So the invention of magnifying lenses led to the discovery of the microscopic world, and it is now known as a single cell which may constitute of the whole organism, as in amoeba, right? It is a single cell. It is the whole organism itself is a cell, and it is amoeba. Its example is amoeba, chlamydomonas, paramecium and bacteria. Now these organisms are called unicellular, that is made up of only single cells. So they are called unicellular. On the other hand, many cells group together in a single cell body and assume different functions to form various body parts, multicellular organisms. So they are multi or many. Okay, such as some fungi, plants and animals. So can we find names of some more unicellular organisms? So we will just see. So what are multicellular and what are unicellular organisms? We will see, and then we will move on to this activity. So all cells thus come from the pre-existing cells. So now we will move on to this activity 5.2. Now in activity 5.2, what we can do, we can try preparing some temporary mounds of leaf peels. We can take some root parts, we can take onion peels or different sizes, cells of different peels of different cells or different particles. We can say that of different parts, leaf peels, tip of roots, onions, or even from fruits and all, we can take some peels. So after performing the above activity, let us see that what answers to the following question we are getting. So what are the questions? Do all cells look alike in the terms of shape and size? So we want to just see whether the cells of all different types of plants, flowers, fruits and all are of same shape or not. So do all cells look alike in structure? So could we find differences among the cells? So these are all questions which we are having in our mind, and we need to clarify that. What assimilates what we are getting, the differences as well as what are the similarities what we could find. So some cells can have, some organisms we are having cells of different kinds. So suppose this is in our body itself, we are having smooth muscle cell. This is like this you can see smooth muscle cell. Then you can see blood cell. So these are all of different shapes and sizes. This is the nerve cell. Now comes the fat cell of our human body. This is the bone cell, this is the ovum, and this is the sperm. So many types of cells are there in our body. So we are multicellular organism. So all the cells which you are seeing in human body are of different shapes and sizes, right? So they are all different shapes and sizes. Now, the shape and size which are related to the specific function they perform, that is some cells like amoeba have changing shapes, that is they are having a body which is very flexible. They can modify or change the shape of their body whenever it's required, right? In some cases, the cell shape could be more or less fixed and peculiar. Like we can say that nerve cells are having a typical shape. They can't change their cell shape also. So each living cell has a capacity to perform certain basic functions that are characteristics of the living forms. Right, so how a living cell is performing certain basic functions? So we need to know that what basic functions is the living cell performing, right? Now, we know that there is a division of labor in multicellular organisms, so each cell is meant to perform different body functions. So, this means that human body performs different functions. So, human body has a heart to pump blood, it has a stomach to digest food, and so on. So, all the organs are specific, having specific functions. So, there is a division of labor for each and every part. So, all work is not done by a same part. So, your liver or your stomach is not doing the function of a brain or a nervous system. So they are having their own part of digestion. The heart is having the own part of circulation. The lungs is having part in respiration. So all are having their individual parts. So there is a division of labor. Similarly, we can say that such a cell also they are having several components. So each part of the cell, the other components are called the cell organelles. So these cell organelles also they perform special function, such as making a new material in a cell, clearing up the waste material from one cell to another, and so on. So the cell also have a specific function. Their organelles are performing specific functions of their own. So a cell is able to live and perform all the functions because of this organelles, right? So these organelles together they constitute the basic unit of a cell. And it is very interesting that all cells are found to have the same organelles, and no matter whatever their function is and what organism they are found in. So they are having the same cell organelles inside that. So we need to answer a few questions before we move on to the next topic, that is who discovered cell and how. So I told you the full basic theory of cell, discovery of cell from 1665 by Robert Hooke till 1940. So what is the full process? You need to just discuss, and how, and what is cell and the structural, why is cell called the structural and functional unit of life. So this is all we discussed till now. So just write these answers in your own way, and if you want to clarify this, you want to ask me any question, write in my comment box. So now we will move on to this. What is cell made up of? What is the structural organization of a cell? So we saw that in the above all the topics what we have discussed till now, that cell is made up of special cell organelles. So how is cell organized? If we study a cell under a microscope, we would come across three important features. That is, in every cell we are having plasma membrane, in every cell we are having nucleus, and in every cell we are having cytoplasm. So plasma membrane, nucleus and cytoplasm are present in every cell. So these are the common things. Now all the activities inside cell and interactions of the cell with its environment are possible due to what? These three features only: plasma membrane, nucleus and cytoplasm. So this parts in details we will be discussing in the next episode or in the next video. So you need to stay subscribed to my channel, so that in the next episode when I'll be discussing with all these main parts, you should get the notification, and you should stay tuned so that you should learn this lesson properly, as I am teaching it in a systematic manner, and it will be easier for you to learn. So stay tuned, stay updated with all my lessons, and if you're having any queries, please do write in my comment box. Thank you.